Myself, Yuvraj Sonone, Head and Associate Professor in Botany, Mahatma Gandhi Vidya Mandirs, SPH, Arts, Science and Commerce College Nampur, Taluka Baglan, District Nashik. Today, I am going to teach a topic anomalous or abnormal secondary growth. Anomalous or abnormal secondary growth. Definition: anomalous or abnormal secondary growth which does not give rise to the normal pattern of single vascular cambium which forms xylem and phloem. Rather, they possesses multiple cambium giving rise to abnormal secondary structures called anomalous secondary growth. Angiosperms and gymnosperms show normal secondary growth, but many plants of these groups show abnormal secondary growth. Such abnormal structure is known as anomalous secondary growth. The secondary growth which gives rise to vascular or non-vascular tissues differing from the usual or normal type is termed as anomalous secondary growth. Causes of anomalous secondary growth. Anomalous secondary growth occurs due to following reasons. Number 1, abnormal position of cambium. Number 2, abnormal behavior of normal cambium. Number 3, formation of accessory cambium. Number 4, formation of extrastellar cambium. Number 5, formation of interxylary phloem. Number 6, absence of vessels in xylem. Number 7, presence of medullary bundles. Number 1, anomalous secondary growth in Bignonia stem results because of two reasons. Number 1, presence of phloem wedges in the xylem. 4 longitudinal furrows of phloem in the form of a cross wedge in quadrants of secondary phloem are developed in the xylem. When the thickness increases, the furrows becomes deeper. Number 2, presence of fissured xylem. The cambium which is in the form of a ring is broken in 8 strips. Out of 8, 4 of them are normal position. Outside the quadrants of secondary xylem and other 4 strips are at the bottom of furrows formed by secondary phloem. The soft phloem tissues being crushed by the pressure of secondary xylem. There are bars of sclerenchyma which appear parallel to the surface of the stem. Number 2, anomalous secondary growth in Raphanus root is as follows: excessive development of parenchyma and lesser development of woody tissues. In the beginning, normal ring of cambium is formed. This cambium activity shows abnormal activity. Instead of forming secondary xylem all over on the inner side, it forms patches of parenchyma alternative with the xylem. While on the outer side, it forms phloem above the xylem and parenchyma at the rest of portions. Thus, a ring of conjoint collateral vascular bundles is formed embedded in parenchyma. In addition to the primary vascular bundles, in the center there is formation of concentric rings of vascular bundles are formed. Number third, anomalous secondary growth in monocot stem, for example, Dracaena. Single layered epidermis which is covered with thick cuticle. The lenticel occurs on the epidermis. Below the epidermis and hypodermis, cork cambium is well developed, which gives rise to the cork towards the outside. Beneath the cork cambium, parenchyma is well developed. The cambium develops in the parenchyma outside the vascular bundles. The newly developed cambium forms cells towards the inside as well as outside. The tissue formed on the inner side of the cambium is differentiated into vascular bundles and remain separated from each other by lignified tissue. The cells developed on the outer side of the cambium become parenchyma. References. Thank you for watching video. Please like, comment, share and subscribe the channel.